got it? Okay. So, yeah, so as John mentioned, I'm not trained in peptide chemistry, although we do a lot of it now. And what we wanted to do at the outset of our research program was build on all of the fantastic advances in peptide chemistry. So, I mean, I think the ability to make quite reliably, 40, 50 mer peptides is an absolutely fantastic achievement. And this is really, I think, to the credit of everybody who came before us. So what we wanted to do was to build these peptides and then cleave them uh, and get the deprotected peptides and then find a way to join those peptides together to make an amide bond. So this is a chemoselective ligation, and this concept is nothing new. This goes back to Kemp and Merrifield and Kent and many other people who proposed this idea and developed some really fantastic chemistry towards doing this. So, I was not a peptide chemist at that time, but we were trained in reaction development, and we liked this problem because it was really one, in trying to develop a new reaction. So what we needed to do was to find two functional groups that we could put on the ends of the peptides so that when we would combine these completely unprotected peptides together, they would make an amide bond. And ideally, this would happen in the presence of all of the unprotected side chains, in the presence of water, without reagents or catalysts. Of course, make a native amide bond and not occur with epimerization. So all you need to do is find the right two functional groups for this and put them on your peptides and mix them together, and then you can make longer peptides and proteins. And there are some solutions to this. So native chemical ligation is a almost miraculous reaction, and it fulfills basically all of these criteria. So we set out to find some other ones, and that led us to this reaction, which we call the CAHA ligation, or keto acid hydroxylamine ligation. It's exactly what it sounds like. We have an alpha keto acid and a hydroxyamine or alkoxyamine, and we mix those together in a polar solvent like DMF or DMSO water, and they join together to form an amide bond with loss of carbon dioxide. And as you will see, this reaction is tolerant of water, it's tolerant of functional groups. It really doesn't care what else is in the reaction. It's sort of like teenagers in love. These two things just stick together and ignore the rest of the world. And just very briefly, there's actually a couple different mechanisms. There's 3 distinct mechanisms that can happen, but all of them involve first the formation, this hemiaminal, which can either directly decarboxylate to give the amide, or it can proceed via the formation of this nitrillium. Okay, so this is a great reaction, but we didn't want to make simple amides, we wanted to make proteins. And to do this, we had to be able to install these functional groups onto the respective ends of our unprotected peptide segments, right? And we really wanted to interface with all of the fantastic operating systems for peptide chemistry. So FMOC chemistry is fantastic. You can buy all the starting materials. Thanks to Roche, everything is quite cheap nowadays. And so, we wanted to use all of this infrastructure, but be able to put the keto acids at the C terminus and the hydroxylamines at the N terminus. And you see a lot of names down here. These are PhD students and postdocs who work 6 or 7 years in really building the technology that nowadays makes this completely routine and transparent. Okay, so do you know if we... So the keto acid, somewhat to our surprise, so peptide C-terminal alpha keto acids are actually really robust, stable compounds. They don't epimerize. If you treat them right, they're really stable. So we just treat them like any other peptide acid. You can do HPLC on them, you can lyophilize them, you can store them for months. There's really nothing special about handling them. The trick is to figure out how to make them. And for that, we had to develop a couple new chemistries. So we developed an approach using a cyanosulfurylide, which we can oxidize to give the keto acid. And we also developed a special protecting group, which upon standard TFA cleavage from the resin, gives you directly the unprotected peptide keto acid. So in order to make this, what we do is we just start from the standard FMOC amino acid. We couple this with EDCI or T3P and this salt. This gives the cyanosulfurylide. These are really robust, stable, perfectly handled compounds. And then oxidize that with oxone to give the keto acid and install the protecting group. So this is now the protected monomer that you can install onto a resin. So we couple it to a resin, and then just do standard peptide synthesis, cleave it from the resin, and you get a peptide, unprotected peptide keto acid. It looks something like that. And we've made this for about 8 or 10 different peptides, or amino acids. For the hydroxylamines, it's a little more tricky. So many hydroxylamines are not that stable. So if you make them, they tend to have some elimination or stability problems. But what tends to be very good is cyclic hydroxylamines. So we have many different types of cyclic hydroxylamines that we have made and employed for CAHA ligation. So we have this oxyproline, which gives a homoserine residue. We have one that gives an aldehyde. We have the serine forming one, although it's quite hard to make. So I recommend the threonine forming one, which is much better. And just recently, we have one that directly gives L-aspartic acid at the ligation site. But it turns out the one that we really like is this 5-oxyproline. It's easy to make. We have an organo-mediated route to synthesizing this. This has been done on a kilogram scale. And it's really robust. It's basically proline. It acts like proline. It has no stability issues. You can do anything that you can do on proline with this hydroxylamine. And the other thing is it has a really surprising feature, and that is this hydroxylamine and only this hydroxylamine actually does not give the amide. It gives the ester at the ligation site. So if you do CAHA ligation, you combine these together, DMSO water. Under acidic conditions, we usually use oxalic acid. And your primary product is the depsipeptide. And if you take that and add some base, just like folding buffer, for example, it rearranges to give you the amide. And it turns out this is really valuable because it allows us to put these peptide segments together, leave this ester bond in there, and it lets us work on very, very hydrophobic, like crazy hydrophobic peptide targets. And so nowadays we preserve this ester usually until the end of the synthesis, and then right before or during folding, we rearrange everything in order to give the amide bonds. So for example, we've used this for insulin, so you can make a A chain with a little bit of the B chain here, linked by this prosthetic C peptide. This was a collaboration with Sanofi. And you make the keto acid B chain. You combine those together. This is what the HPLC of the ligation looks like. So that's the product forming. It goes overnight to give the major product here. We purify that. And then we remove the ACM groups and fold it. And during the folding, we do the rearrangement to the amide. We cleave out the C peptides, and we form the disulfide bonds to give. This is the major product here, which we can isolate to give these insulin variants. And the other thing that we're interested in is therapeutic proteins. So things like cytokines and chemokines. So this is very robust technology, and we can make a lot of therapeutic proteins, up to about 200 residues, using 3 or 4 segment strategies. What's really nice about the CAHA ligation is that we can orthogonally protect either the hydroxylamine or the keto acid. So we can assemble in any direction we want. For example, here we use the photoprotected keto acid. We do the first ligation, and then in situ UV irradiation to give us this keto acid segment, which we isolate and purify. Then we do the next ligation, and then in situ rearrangement to the amide bonds, and then ACMD protection and folding to give, in this case, IL-4. We've done this for a number of different things. The co-ligation goes under, it doesn't have any additives or things like that. It's very easy to scale this up. And we have prepared hundreds of milligrams of the linear proteins, and we're working now on basically a 10-gram scale for making some of these proteins. The folding yields are not quite as good, but they're still good enough to get substantial amounts of these kind of proteins. And they all look very nice. This variant of IL-4 does have 2 homoserine residues in it, but it's equipotent, actually a little more, and in fact, than the recombinant material in standard assays. I don't think it's actually more potent. I think the recombinant material is just not that pure. So many proteins, many different types of proteins, very hydrophobic proteins like IFITM3. We've made cyclic proteins. We've made beta sheet proteins. So you can access most things, again, up to about 200 residues using CAHA ligation. And so still the hardest part, the bottleneck, is still often making the peptide segments. So once we have an optimized sequence, this is we routinely make things on 500 milligram scale in our lab, even to actually find the right sequence, especially for some of these very hydrophobic proteins we're making, like cytokines and interleukins. So we're fortunate to have 2 peptide labs, one in Nagoya, one in Zurich. I didn't know anything about peptide chemistry at the beginning, but having these 2 groups working on peptides let us make twice as many mistakes. And so we really learned a lot about how to do this. And the one thing that we learned, I think the most important thing, is that peptide synthesizers are monogamous. They only like to work with one person. And so it's very important that you have the right technicians working with the right synthesizers and keep them running 24/7. And so I really want to thank the technicians that we've had over the years for really turning this into a little protein factory. Okay. Oh, let's go back. Oh, yeah. So one of the things we learned the hard way is that one of the biggest problems in making very long peptides is aspartamide formation. And so here's a notoriously difficult sequence. It has this DN and DD groups here. And if you just do standard FMOC chemistry, this is the crude HPLC. And unfortunately, this is not actually the product. So we've suffered from this. I think many of us have suffered from this. And what we found actually is that we can use these cyanosulfur ilids as protecting groups for carboxylic acid. So this is a carbon-carbon bond. It cannot form aspartamides. So we made this derivative of aspartic acid, and when we use this instead of the O-tert-butyl derivative, this is what the crude synthesis looks like. This is just standard FMOC chemistry, nothing special, no double couplings, just normal, and that's the purified one. And then we have to remove this group. And we do this with NCS. It's actually a very quick reaction. So this is the starting material. This is the crude peptide after being treated with NCS at pH 3 in water. That's the purified one. And then we can, in this case, remove the ester-butyl groups and fold this. And the mechanism is quite interesting. So what happens is that this group gets chlorinated, and this is basically like a haloform reaction. So this ketone is hydrated, and it breaks this carbon-carbon bond to return this. And so the NCS actually, almost everything is tolerated. Methionines are not tolerated. But tryptophans, tyrosines, all of those kind of things are perfectly fine under these conditions. So I think the advantages of the CAHA ligation is the conditions and the chemistry. It's really perfect for hydrophobic peptides and proteins. And it's convergent, so we can do things in any order because we have these orthogonal functional groups. We can even combine it with NCL, so you can use the thioesters under the CAHA ligation conditions and take that directly into NCL reaction. And we get these esters, which is often quite valuable for these hydrophobic targets. So I guess the limitations, so one is that if we use the 5-oxyproline, we get this homoserine residue. So for most of the chemical biology projects, even the therapeutic protein projects, this is not a big issue. But of course, if you're making an API, then this is not going to be acceptable. And so we do have some alternatives, and threonine and aspartic acid are the best ones. And you do need these special starting materials. So again, these have all been made on kilogram scale. We have suppliers to do this. They're not catalog products at the moment, but they may be someday. But once you get your hands on those, then it's really easy to do this chemistry. Okay, so what we would like to do and what we're very interested in doing is protein-protein conjugation. So being able to take two different proteins and combine them together, ideally keeping them in their folded form. So we would like to do, for example, ubiquitination. This is an example where we tried. This is synthetic ubiquitin with a keto acid. This is IFITM3 with a hydroxylamine. But this reaction doesn't work. For one, we have folded proteins at pH 2. This is fine for some proteins, but not all proteins. And then, chemistry is just slow. So the CAH ligation is not a slow reaction by organic chemistry standards, but it's still, not fast enough to work with reasonable stoichiometry at micromolar concentrations. And this is, I think, a real problem with chemistry, is that when you get to folded proteins, you're at micromolar concentrations, and most chemistry is simply not fast enough. So what can we do about that? We've developed new chemistries. So we have a kind of variant of the CAH ligation that we call CAT ligation, which uses potassium acyl trifluoroborates. These are funny-looking functional groups, but they're actually very, very stable, very easy to handle. And this reaction is about 1,000 times, 100 to 1,000 times faster than the CAH ligation. So we can do things like protein pegylation and protein-protein conjugation using that. And you can also do some really fancy things. So you can actually make derivatives of these acyl boronates and put a biotin here. You can do the same thing with the hydroxylamine. And then you can do traceless templated ligations. So we can take these with streptavidin. And the reaction itself is not super fast, but the templation gives it an effective rate constant of about 200 per molar per second, which is very impressive. And so you can use this to join things together. This is not yet, we haven't yet done this with proteins, but there's a nice proof of concept study that was published this year. But this is still a lot of work. It may be possible to do this, but you have to put a lot of effort into installing the functional groups. And that's not necessarily what you want to do, especially if you're working with recombinant proteins. So it is still really hard to do this, And I guess one consolation is that it's actually hard for nature as well. So nature does protein-protein conjugation in the form of ubiquitination, but it uses a very complicated process involving 3 discrete steps. It has thousands of enzymes just for doing this job. And roughly speaking, what happens is you have a protein like ubiquitin that gets activated, and it couples with an E1 enzyme that transfers to an E2 conjugating enzyme. And then with the assistance of an E3 ligase, it binds, you can't see this color here, but it binds to this target protein and transfers the ubiquitin. And we were particularly interested in one of these E2 conjugating enzymes called UBC9. This is the one that canonically does not transfer ubiquitin, but a ubiquitin-like modifier called SUMO. And the interesting thing about UBC9 is that it can work without an E3 ligase. It's not fast, but it does work. And it actually directly interacts with the substrate, which is quite unusual. And it has a consensus sequence, which is a hydrophobic amino acid, a lysine, and this glutamic acid. And it likes to transfer things there. And so, it's known that if you take an E1 enzyme and ubiquitin and a substrate protein like RANGAP1 and UBC9, and you mix them all together, you can get very clean ubiquitination. And so the dogma is that you need the whole folded SUMO in order for this process to work. But we're synthetic chemists, so we thought, what's the difference between a peptide and a protein? Let's just try the peptide. And so you can make the C-terminus of sumo, which looks like this, and you can try it. And unfortunately, you can't see it on this projector, but there is a band here. And so it does work. It doesn't work very well, but it's not zero. So we thought maybe we can improve this. But the first thing we wanted to do was make sure this was not a background reaction. And so we took something which is known not to work, which is ubiquitin, right? So we took the ubiquitin sequence. Where is that? And surprisingly, if you take the ubiquitin thioester, then now it works quite well. So this is quite surprising because it's not supposed to work with ubiquitin, it's supposed to work with sumo, but the ubiquitin peptide is the one that likes to conjugate. And in fact, you don't even need the whole ubiquitin sequence here, so you can chop it down, and basically the minimum sequence that you need for decent reactivity is just this gly-arginine here on this thioester. So this was pretty cool. And so what we did is we took this tag out of RAN-GAP1 and we installed it on the C-terminus of GFP. And that also works very nicely, so we can take UBC9 and one of our ubiquitin-derived peptides, and that transfers directly to that one lysine and only that one lysine. It has a half-life of about 60 minutes. And this is proceeding at 15 micromolar protein concentration. And then we did an alanine scan. This is the whole tag that we took out. And basically what you see from this is that you need the lysine, of course, you need the glutamic acid, and you need some kind of hydrophobic residue here. But all of the other residues, it really doesn't care about. So you can mutate those to whatever you like. So this is sort of the minimal tag. We call this LACE, lysine acylation with conjugating enzymes, and it's this 4-amino acid LACE tag that you can use for site-specific conjugation to proteins. So you need a couple of these residues, so you need these 2 arginines, or at least this 1 arginine, but after that you can have whatever you want. So you can have longer peptides, you can have dyes, you can have biotin, and what we like to do is put our functional groups for our cat ligation onto proteins using this technology. You can put this tag more or less wherever you like. You can put it at a C-terminus, you can put it at a loop, you can put it at an N-terminus, you can put 2 of them on a protein and do dual labeling. We can put it into different recombinant proteins like this IG domain from Titin, lysosome, and we can put this into FAB fragments. And what's very cool here is we can put a sortase domain on one side, and we can put a LACE tag on the other. And then in one pot, we can use LACE to attach one group here, and then in the same pot, we cool it down and add sortase and transfer something else. So this really works to do dual labeling of FABs. Everything is recombinant here, and you just mix everything together. Okay. So if it's the ubiquitin sequence, so in principle, this should work for ubiquitin as well. And so what you can do is you can express ubiquitin as an intine and cleave it to get the thioester. This is really easy. You get like buckets of this stuff, like 100 milligrams. So it's sort of annoying to purify on our GPC. So you get this ubiquitin thioester, and then you just take UBC9, and on this tag, you can monoubiquitinate. And so here you can see this gel. So it's not super fast, but after 48 hours, we have about 70% conversion. Just one ubiquitin is transferred, right? There's like 20-something other lysines here, but it's only hitting this one where the tag is. So it's quite specific. So what you can then do is you can go put this tag into other proteins. So this is sumo. It has a native sequence with the valine here, but LACE likes isoleucine better, so we make that 1-point mutation, and then we can put ubiquitin onto sumo. We can do the same thing on synuclein, which has 15 lysines, so we make that 1-point mutation, and then we can put ubiquitin specifically onto that lysine, or we can mutate this glycine to isoleucine, and now it transfers to the 102 position. So this is monoubiquitination, on recombinant proteins, with exquisite specificity. So we were, wondering how this works and why this works with ubiquitin rather than sumo. So it turns out the rate determining step of this reaction is not the amide bond formation. It's actually loading the thioester onto the E2 enzyme. So we thought, okay, surely, UBC9 is just slow to react with the ubiquitin thioester. It'd rather have the sumo thioester. But that's wrong too. Actually, it's really slow at loading the sumo thioester, and it much prefers the ubiquitin thioester. And then we thought, well, surely the transfer with the sumo thioester has got to be faster. This is the natural substrate. But that's completely wrong too. It's also faster with ubiquitin. So bizarrely, UBC9 really likes ubiquitin, and it doesn't really like its native substrate. So that was baffling. So what we did, or I should say my student, Raphael, who is amazing, what he did is he went and we tried to grow we made this synthetic peptide, and we kept getting apocrystals, but then he put this cysteine in here, and this made this disulfide link, and we could get a crystal structure basically of this peptide here. This is from the donor, and this is from the lace tag of UBC9. And so you can see basically how each of these parts are fitting into UBC9. And so I don't have time to do the full analysis here, but here's the lysine residue, right? And so this is the amide bond that was formed. And what's amazing is that this unnatural ubiquitin substrate fits perfectly in. There's this hydrophobic pockets where the leucine residues, which are not present in sumo, fit, and then there's this acidic patch here where the arginine residues bind. So it's really a perfect fit for this. So, so I think I'll stop there. So we have two technologies. One is this LACE technology. This is only the second time I've talked about it, so I would welcome your feedback. And what we can do is with complete